I have told you a number of times, if you've been here before, how important an inaugural lecture is. And in the past, I've talked about how important inaugural lectures are for those people who've been here a long time and they've gone through and their career is being based here at the UCL School of Pharmacy. Inaugural lectures are just as important for those people who join us. And I think that this is my 4th School of Pharmacy, and there aren't that many people who are female and work in the area of drug discovery. So it's really great that we are celebrating not only Zoe, but Zoe as a role model for female colleagues who work in drug discovery. I also feel, as you can tell, completely underdressed for the occasion. She did tell me she got something special. So it's a real honour that actually Zoe's asked me to do the welcome. So just a few things that you may not have heard about Zoe. So first of all is the fact that she came here 5 years ago. If you wind back, you will remember that there probably wasn't a lot of people to say hello to Zoe when she joined the UCL School of Pharmacy from UEA, because obviously we were in the pandemic. So it must have been quite challenging for Zoe to join what we think is a vibrant and inclusive community here when actually we were all sitting at home. So Zoe, no doubt you'll have some reflections to talk to her about that as well. You will also know that research is really important, as is education, for being a professor. And Zoe's research and interest in the area of DNA, particularly related to insulin, was the thing that brought her here. But also, I think she'll also mention that actually it was somebody else who brought her here. And it was that telephone call. I know I was there too. When you get that telephone call that says you'll probably be very busy, but you might want to know that there's actually a post coming up at the School of Pharmacy. Just thought I'd send you an email. Happy to talk to you in case you might miss it because you're a very busy person. So that would have been Duncan Craig, because that was his way of making sure that people were aware of that, as well as obviously advertising, as you would expect in the right way. So no doubt Zoe might talk about that, especially as I know that Duncan was actually at UEA as well. Zoe also is a passionate educator. So I think you'll learn more, and she may well use this as a conduit also for education. We have some small people in the audience who she might be directing some of that to as well, to follow in your footsteps perhaps. But also, being a professor is not just about education and research. It's also about external engagement and institutional citizenship. So I'm hoping that Zoe's going to talk to you a little bit about the external engagement she's done, particularly with the Royal Society of Chemistry. I'm hoping she's going to allude to that as well. She has more than one role in that area. And also institutional citizenship. So it wasn't long before Zoe joined when she actually stood up and said, I'm happy to be your Associate Director for Research. And I thank her for that, because I think she's done an amazing job. And those of you in the audience who are now members of PBC, the Research Department of Pharmaceutical and Biological Chemistry, are hopefully also pleased that she stood up more recently to become board. So we want to thank you for that as well. So I hand you over to the very glamorous Zoe Waller to tell you about her story and what brought her here. Over to you, Zoe.
Hey, wow, and thank you so much, Kate, for your very kind introduction. I'll just make sure that I am doing everything correctly, because there's technical stuff to do as well. So I'm going to share my screen and just want to check with the folks online that they can see that okay. Anyone can give me some indication in the chat, that would be great. We can see your slides. Good, good. Thank you so much, Andy. And there we go, I'll close that. And I'll put that to full screen. And then hopefully that should all be working. So thank you so much for coming. It's a little bit overwhelming. And thank you again to Kate. And I'm going to talk to you today about the changing shape of DNA. And essentially this is going to be I don't know, I guess a bit of show and tell really, of my story. And I did I did wonder about what order to do things in, but I've tried to work out what the most rational order is, and sometimes things aren't quite in chronological order to make it a bit easy. So I'm going to start at the beginning. Oh, please don't tell me this isn't going to work. There we go. So I was born in 1983. This is me. And these are my parents. This is my mum and my dad. And we did have some questions about who this was. I did find the other photos to confirm that I'm in the photos of my brother Joel. So this is me. And I was brought up in a place called Ipswich. But I would talk to my dad about this. They did a lot of reflection of like my journey. And then he said that my mum was, unfortunately, she was quite an unwell person. And he said that it was the best he saw her when she was pregnant with me. And the reason I mention this is because my mum and her illnesses affected a lot of my journey. And then it was one of those things that has influenced me a lot. So, Dad. Of all people. So, whilst my mum was unwell, I was also raised by my family. So my maternal and my paternal grandmothers were really key to my upbringing. They were very strong female role models. My dad was one of 5 children, and I had 4 aunts. And out of all of my cousins growing up, all bar one, whilst I was growing up, were female. So I didn't know what the matriarchy was, but it definitely was part of my upbringing, even though I didn't realise it till later on. So they were very, very strong role models. Even when mum was ill, there was always plenty of people around to be a strong role model for me. And another part of the influence is, so this is BT Research Laboratories based in Martlesham Heath. And we lived in the village just opposite, which is basically where everybody from BT lived. And the reason I'm telling you this is both my parents were technicians. They worked in photonics, and they're actually involved in the team that made the fibre optics cable that ran under the Atlantic, part of that team. So from a very early age, I had a very strong science influence. I'd never had a chemistry kit, but I made my own dyes. Like my mum and I once made dye out of like onion skins. Didn't really work out too well, but we worked out, we liked the chemical ones a bit better. And things like a very early understanding that if I didn't get the pen off my hands with some soap and water, I could go and get mum's nail varnish remover and use that instead. So there was a lot of science very early on, although it was kind of endogenous rather than being taught it specifically. The other influence is that both my parents were quite artistic. So my dad is still an organist, so he did a special event in this. My mum was also very artistic, and she herself was influenced by her uncles, who were both 2 of her uncles who are artists. So my piano teacher told me that she thought that I was brought up in a Renaissance environment, and I didn't know what that meant when she told me it was, but I appreciated it when I started learning it in art at the age of 14. So then the other thing that I would like to show you is some of the things that I have done along the way. So this is an example. So this is me when I was 11. Yeah, I know. One of my tie-dye tops, and this is an example of some of the things that I used to do. So I used to make quite a few things, and this was something that I made in school. And I'm at that age where my parents, my dad's been like cleaning out the house and finds a load of stuff. This is yours. So I've been quite lucky to be able to have some of this basically propelled into my direction. So I have been using this slide in quite a few years in some of the talks that I do in schools, which Cate has mentioned. And one of the reasons I bring this one to is because it depicts essentially what I wanted to do when I grew up. And I actually wanted to be an animator for Disney. So it's not going too well so far. I was really keen on art, and I had that big influence from a very early age. Obviously, something changed as I went along. And when I went to high school, so this is a picture of my high school. When I showed this to Pip last night, yes, it looks really grim. I don't it's true, though, right? Like, so it was a nice school. It was a nice school. And then there I learned science. So I did get to see some of my science books. I don't know what that is, but I thought it was important to stick it into my science book. And it was a really, really different environment. So at primary school, I don't even remember learning science, but at high school, I definitely did, and I very much enjoyed it. And two other things kind of came whilst I was a teenager. My best friend at school, Rebecca, her mum was a science teacher, so she always knew a little bit extra, and we nerded out together. I think we've both done all right. She's a consultant in public health in Essex, and so like we used to do this. We used to be part of a science club, and it was basically just us. So I'm grateful that we had this together. The other influence was this person here, which is Gillian Anderson or Daniel Scully from the X-Files, and it was a massive influence on me as a teenager, and I very much am a product of the Scully effect, which helped bring people into certain subjects. And although how obsessed I was a little bit, well, probably, I'll show you. So this is basically my bedroom as a teenager, and I collected everything, I drew things. The X-Files infiltrated every part of my life, including my art. And this was actually, I won a prize for this in a school competition, but every part of my life I thought about science. And I wanted to be, essentially, I decided that I wanted to become a forensic scientist. I wanted to be just like this person here. There was only one problem, though. I wasn't too great at maths. So this is my end of year assessment for year 10, and I don't know, Eddie's done percentages this week. But 8 out of 56, you don't need to work out the percentage to know that that's not very good. And actually, my teachers tried to advise me against choosing science A-levels because they didn't think that my maths was good enough. Now, I've worked out in subsequent years when I did my teacher training that actually I probably, although I'm not diagnosed, have a thing called dyscalculia, where you get the numbers mixed up. And actually, I ended up, in hindsight, putting in steps that helped me with my numbers. So to get over this, I ended up doing a lot of background reading, back work. I feel like my maths GCSE was the only one I revised for, and my dad helped me a lot. So thank you, Dad, for helping me in any way. And the other thing that helped with my maths is I worked in a fish and chip shop, and that was really good for my mental arithmetic, which needed it. So in 1999, I got my GCSEs. I got 1A, 2 Bs, and 7 A stars. And one of my Bs was in maths, but I was really pleased because until the year before, they were a bit concerned that whether I was going to make the grade to go to A-level. So I was super pleased with that. And although the level of GCSEs wasn't like all A's, something to be clear is that I got the joint highest grades for GCSEs, putting in context for that school. After that, I went to sixth form because our school at Kesgrove didn't have a sixth form, and then I went to Northgate High School to study biology, chemistry, physics, and maths. And actually, when I made my A-level choices, I was supposed to be doing biology, chemistry, history, and geology. And then I walked into my first history lecture lesson, and I didn't like the teacher, so I quickly swapped to physics and maths, which I knew that if my teachers, because this was after I'd left school, I knew my teachers would have advised me not to do that, but I got my B, so I could do both of them. And sixth form was a really interesting time. I met lots of new people. So James Williamson is somebody that is also a scientist, and also he works based in Cambridge now. And one of the other people I met at that time was Philip, who was there tending to the, yeah, that's Daddy, tending to the children. So I was, as you can probably see, like my bedroom was a complete state. There was a lot of patterns. It was the late 90s at this point, the early 2000s at this point. There were a lot of stuff stuck on my wall. One of those things, ironically, is my revision for transcription and translation. So I think that's come in handy along the way working with DNA. I didn't know what I wanted to do at university. I had a very strong influence of that. Going to university, I didn't know what I wanted to do. I knew I wanted to do forensic science, and there was advice to do a strong, core, solid subject, not doing don't do a forensic science course, do a pure science and then apply it. And my UCAS form was a complete mess. So I think some of these was like masters in biochemistry, masters in science, chemistry. I think this was analytical science, this is biology. So I wanted to work at the interface between chemistry and biology. And at sixth form, because mum was getting quite unwell, so in addition to having bipolar disorder, she also had multiple sclerosis, and ended up sharing the caring for her with my father as well. And that did impact on some of the work and what I was able to achieve, but I wouldn't have swapped it for the world, because the amount of different transferable skills I learned from managing my time and things like that as well. In addition to all of that, I also got a job in the library. I worked alongside Pip and also my now mother-in-law, which was interesting at times. And because I learned a lot about the menopause working here. All of the people working in here were female, and there was just one topic all the time. So I learned a lot about the menopause. Obviously, Pip does as well. So hopefully we'll remember some of that soon. Be realistic. And then I got my A-levels, and then I chose to do a degree at the University of Bradford in chemistry and pharmaceutical and forensic science. And it didn't look as nice as this when I went, but that's what it looks like now. And I was super pleased to go to university, but I did what the teachers did not advise again. They said, don't go to university with your boyfriend. That's not what you want to do. And I don't like being told what to do. I've realised this over time, but I went to university with Pip. I think it worked out okay. And whilst I was there, I met Vicky, who also was idiotic enough to go to university with her boyfriend, and Philippa, and together we were referred to as the Three Musketeers, because we were the nerds that sat at the back but always had our hand up when answering any questions. And the other person that we usually sat with was Andy, who played rugby with Vicky's boyfriend. So we were kind of a four together. So Vicky and Andy did their practical classes together, and Philippa was my lab partner. Now, Philippa and I shared a lot of loves for different things. We loved the same type of music. She also loved interesting clothing and jewellery. And I think my first words to her were, I love your bracelets. But the key thing was that all three of us had a real love for science and also really wanted to do something good. And so we'd chosen the forensic science course for that, this chemistry of pharmaceutical and forensic science. Now, I wasn't particularly interested in pharmaceutical science at that point, and I thought it was going to be really boring. Turns out it was actually the bit that I found far more interesting than the forensic science. Philippa was really interested in pharmaceutical science because she had cancer when she was a teenager, and she wanted to, her career aim was to help people with cancer. And unfortunately, in our second year, Philippa had back pain for like the majority of the year, and she had regular checkups because she'd formerly had cancer. And then she had a diagnosis, essentially, of having cancer in 6 different parts of her body. And unfortunately, within 6 weeks, we lost Philippa. And I don't think anybody expects to lose somebody that they're so close to so quickly at the age of 19. And it just made me completely reevaluate everything that I wanted to do. This is Philippa and Pip, and this is me and her. Again, notice got all of my notes on the side there. So I had a lot of thinking to do. And in that summer of 2003, I was working in this place, which, if you think that looks grim, it was pretty grim. This is County Hall in Ipswich, and it was formerly used as a prison. Apparently there's an auction tomorrow if you want to buy it. It's being redeveloped into flats. But I was in a windowless room, essentially organising training for people in the library service. And I spent a lot of time in my lunch hour, an actual hour for lunch that I actually took and sat and ate my lunch and looked through the internet, and I was looking about cancer, about how to cure cancer, essentially, naively. And I learned about telomeres, and I did a lot of reading. And I decided that I didn't want to be a forensic scientist anymore. I wanted to do a PhD. I wanted to do something to fill it up. So I learned about telomeres, and also in 2003, I learned about telomerase, and I learned and found out there's somebody at Cambridge working on telomerase, and that person is the now Sir Shankar Balasubramanian. And I decided that I wanted to work in this area, and that's what I wanted to do. And I was really lucky because I had a fantastic mentor, Professor Richard Bowen, who's here today. Shankar's also here today, but thank you for coming. Richard was like super supportive. He was really keen to see me go to Cambridge, and he supported me with advice, and he offered for me to work in the lab in the summer. Unfortunately, because of potentially looking after mum, I wasn't able to take some of those opportunities. But I also learned a lot more biology because I knew I was going to need it. So then in 2004, I started applying for PhDs. This is the Department of Chemistry in Cambridge. And I applied and I got interviews for at Cambridge with these people here. And then I was so super excited. And then I arrived at Cambridge for my interviews. And then I found out that Shankar was not there. And he sent his apologies. And I did my interviews, and I got some offers for some studentships, and it didn't sit well with me because I just felt like everything had gone wrong, even though it was silly because I'd still got an offer from Cambridge. So I was actually in an RSC lecture that we were attending as undergraduate students, and I just decided I'm going to send him another email. So I sent Shankar another email and said, oh, I'm really sorry to miss you. I really wish that I could have had the opportunity to meet you. And he said, yeah, sure, come in a couple of weeks' time. So I went back and saw Shankar, we had a chat. And he said, oh, yes, so I'd be happy for you to do a PhD with me. And I was like, yep, okay. And then he's like, do you want to think about it? And I was like, no. I'm sure this is absolutely what I want to do. So I don't think Shankar knew that I knew about him for 2 years before I sat in his office. But I know this. Not stalking, I wasn't stalking. My PhD was going to be on chemical genetics. It was a project that was funded by the BBSRC, and it was going to be targeting G-quadruplex DNA. So there is this relationship between quadruplexes and the telomeres, and I will explain a little bit about that. But before I explain a bit about DNA, to finish off my story about Bradford, I did my MChem in a project that was a bit analytical science, a bit synthetic, so there was a bit of two different things. I really enjoyed my project. It was with Professor Richard Bowen. I absolutely loved being in the lab. I loved doing things with my hands, and I think that kind of relates to my artistic nature, is that I love doing things with my hands. And this picture was actually taken. Okay, I know I have goggles on. I'm aware that I'm not wearing a lab coat. It's a white jacket. Not covering my midriff. But this was literally my now in-laws were sat outside in the car with all of my stuff from Bradford packed. This is me. I'd finished. I'd already done, like, I'd submitted. I'd done everything, but I was still so interested in finishing off laboratory work that I'd volunteered to go and make sure that this was dropping at the correct rate before I went home. So we adjusted this dropping funnel to make sure it was going at the correct rate, adding that notes there. Working with Richard was amazing because I learned so many different things, and I'm super grateful for his mentorship, continued through my career. So I graduated from Bradford, and then here's Richard here with me and my other supervisor, Howell Edwards. And I was really lucky that I'd done enough work to get a paper out of it. So we published that in the year after I graduated as well. So then I arrived in Cambridge to do my PhD in chemical biology, and I was super excited to come and work for Shankar. Cambridge is a very, very odd place, and I hadn't clocked how different it would be from Bradford, especially because this is Peterhouse, this is the college that I was in. But I was there anyway, and I came to work on targeting G-quadruplex DNA, so special types of DNA structures using chemical genetics. Now, I think it's helpful to talk a little bit about DNA, so we can all be on the same page. DNA is an information molecule. It codes for all life on this planet, from Goliath, our cat, to, as I was growing up, to the tomatoes that Pip was growing in the garden. All life on Earth is encoded in DNA. So all our bodies are made up of different types of cells. In the cells is a compartment called the nucleus, and in the nucleus, our DNA is held in the form of chromatins. This is our DNA here. And DNA is made up of 4 building blocks. We sometimes call these the building blocks of life: G, C, A, and T, guanine, cytosine, adenine, and thymine. And these pair up, they base pair, so they're like friends. So guanine likes cytosine, adenine likes thymine. And the genetic code is essentially these sequence of bases. And the humans have a genetic code that's 3 billion bases long, and all different organisms have different sized genomes. So you can imagine E. coli is quite small, whereas it might come as a surprise to you that a newt has a larger genome than a human. And I keep telling Pip this, but plants have the biggest genomes. I should put one there. I did look up at this rhododendron behind me, but the rhododendron is approximately the same as humans. It's not bigger, but the largest genome, I think, is 160 something billion base pairs. So plants are very special. I've got some agreement from our plant science people here. And so what does DNA code for? So DNA is like a blueprint or a master copy. And then to use it, you need to make a working copy, which is called RNA, and then that gets made into protein. And I liken it to, so imagine you've got a first edition of Mary's Berries, Cakes and Bakes. You probably don't want to, and it's a signed copy, you probably don't want to do all of your baking, especially if you cook like me and everything is everywhere. So you might want to take a copy of it, either a photocopy or a photo on your phone, and then you would use that to then make your cakes. DNA is inherited, and you probably know what this is. So this is a picture of me when I was about 18 months old, and you'll be able to see through each of the individuals. So this is Eddie, this is Griffin, and this is Franklin. Franklin doesn't have the same smile as me, but the other two do. But we all know that DNA is something that you inherit from your parents. Yes? Where's Griffo? This one's you, darling. Yeah? Yeah, good job. I know they all look the same. It's going to be fun 18th birthday parties. Which one is it? Sometimes people say your son has your eyes, which is nice, and Franklin does have the same colour eyes as me. What they should actually say is your son seems to have inherited a gene from you that makes the protein in his eyes appear the same as yours. It doesn't quite have the same ring to it. Franklin doesn't care anyway. So what does DNA look like? And this is where we come into the structure of DNA. So DNA, I like to think of it as shaped like a twisted ladder. And this is the structure that was first proposed by Watson and Crick in 1953. There is some controversy around that. And if you're interested in the history of DNA structure, Professor Stephen Needle wrote a really lovely article for the 70th anniversary of the structure of DNA about Rosalind Franklin and the situation, which I'd highly recommend reading. But this is essentially the structure that was first proposed in 1953 and then later proved by crystallography in the 80s. In 1962, it was apparent that if you had a lot of the base guanine, so a lot of G's in it, it could do something different, and it would form something slightly different. And this is how guanine, or the G base pairs with it. It can also base pair or pair up with itself. So if we take that guanine, and then I like to think of it like a corner. So you've got two kind of complementary sides here. And then so you can add another one, and that base pairs nicely here, and another one here, and another one here. And if you have a long sequence of nucleic acids that contains these bases, they will actually stack together or form helices to form something that we call a G-quadruplex. And these are composed of Hoogsteen hydrogen bonding. So this is our first kind of indication that G-rich sequences can form G-quadruplex, or an alternative structure of DNA. Similarly, C-rich sequences can also form different structures, and these are i-motifs, which are four-stranded structures that form in C-rich sequences. And they were found in 1993. Some notable mentions because of the significance and to indicate some of the work of colleagues that you will know and love. So in 97, there was a really nice paper that showed that you could stabilize one of these special DNA structures with a compound, and that can affect telomerase. And telomerase is an enzyme which is overexpressed in cancer cells. So 85 to 95% of cancer cells will have too much telomerase, and it essentially makes those cells immortal. This is one of the reasons why I was interested in telomerase for cancer research. In 2002, these four-stranded structures were shown to, the actual structure was shown using crystallography, and that was also work done by colleagues that are present here, Professor Stephen Needle and Gary Parkinson. And then in 2003, the Human Genome Project was coming to completion, and it was very clear that not all DNA codes for protein. And the big question was, well, what does the rest do? And that's where a lot of the research that we're interested in comes in, is that non-coding nucleic acids, the non-coding DNA. And then in 2005, which is just before I started my PhD, Shankar was working with Julian Huppert to show that actually if we look at the sequences that can fold into these special structures, where are they in the genome? And they found that 43% of all genes contain a DNA sequence that can form one of these special G-quadruplexes. And then later, after I left Cambridge, Shankar's group went on to visualize these in cells, and that got a lot of attention from the news. And so hopefully I can convince you that DNA is more than a double helix, and that it can form these alternative structures, the G-quadruplex and also the i-motif. And why we're interested in this is that we can think that if we change the shape of DNA, we can potentially switch genes on or off. And they have potentially a native regulatory function. We can also target them with small molecules or drug-like compounds. And what that means is that essentially we're affecting this pathway here, so going from DNA to RNA. So if you switch on a gene, you end up making more RNA, and that means you can then make more protein. So that aside, that interlude on DNA, this was my PhD working on these special G-quadruplex structures, and this is a comparison. This is actually a drawing that I did for my PhD thesis, which I did by hand. Thankfully, Philip was able to scan them all in and clean them up yes. What's that? That's just told you that, Griffin. That is a G-quadruplex. Any other questions? What's that there? This is called a G-tetrad, because guanine likes to do stuff with itself rather than cytosine. All good? Yeah. Awesome. So Cambridge is. I felt like I had 3 lives. So I had a lab life. Yes. What's them? This is Mummy. Look at that hair. Look at that hair. Wonderful. Posing in the lab. So there was lab life, and we were like, it was very privileged to be in such a great, fantastic group, and so much to do. I was so excited about being at Cambridge, so excited to be working with Shankar, and I did as much as I could. And so these are oddly, was just like this lovely picture of us all together. We did not coordinate at all, but when I was looking at it, I was thinking, we look pretty coordinated for that photo. So we had lab life. We also had college life. So I ended up being the treasurer of the MCR, which, given that I could barely count, was quite funny. And then I made a lot of friends. And one of the things that the porters in college ended up being like multiple extra dads, so it was really, really nice. And then home life, I was on Pimp My Ride, which is a thing at the time, but I know it's a bit mad, though I think Tim Westwood's been cancelled since. And the other parts that were part of my home life was that I went to a lot of concerts and did a lot of gigs and all ran behind the scenes at a lot of the gigs as well. So 3 lives. It got a bit complicated at times, but I think Cambridge has this work hard, play hard ethic, and I definitely did both, well, everything. And in 2008, I had my first paper, which is of these compounds, which are triaryl pyridines that targeted G-quadruplex. And then in 2009, I submitted the paper that eventually went into JAX, which was some more triaryl pyridines that actually were able to switch on gene expression in a particular gene. And it was an odd time because I submitted my JAX paper, and then I wrote my thesis, and then I submitted my thesis, and then I got the comments back from my JAX paper, and then there was a period of months between me submitting my thesis and my viva. And in that time, I disproved chapter 2. I was a bit of a state, and then reproved it. And then I think it was about like a week before my viva, Shankar said, "How's revising going?" And I was like, "Oh yeah, I should probably revise." So then I spent, I think, 4 days just cramming, and my viva was not a good day for me. There are no photos, so I have no photos of my viva. I think that, I don't know, my examiners were Stephen, it was a great day, and David Spring, and the amount of times I said, "Sorry, I don't know," because they told me I passed before I went in, which was a mistake, because I was like, "Oh, I'm good then." So one of the questions Stephen asked was, "How does telomerase work?" I'm like, "Yeah, of course I know how telomerase works," but I said I don't know because I felt unconfident. Yes, darling? Oh, in a minute. Okay, yeah. Yeah, so I obviously knew how telomerase works. I said I don't know, and then I said I don't know to a few other things. And David Spring at one point said to me, "Zoe, I've seen you teach this to Part 1A students. Please just draw the mechanism." So I had to do that. So it was fine. It was not a good day, but thankfully, Stephen's very polite, and he said the candidate showed a good knowledge of. So I got my PhD. It was fine. And then after I finished my PhD, I did get married. Because I was leaving academia, I was due to become Dr. Bradbrook. And one of the things that happened during my PhD is I like to work with my hands, but I'm quite clumsy. And when you're quite clumsy, you break a lot of glassware. And then when you break a lot of glassware, you tend to get friendly with the glass blower. So I have Keith here, who's the glass blower at Cambridge. And he fixed a lot of stuff for me during my PhD, but we're good friends. And so he was my best man when I got married to Pip. And he made this beautiful lab coat for me that said Dr. Waller, and it said for one night only, because I was like, Cambridge sends you this information that says you should not call yourself doctor, you cannot do this. And I was like, oh, someone's going to find out. But no one did, no one cared. And I'm Dr. Waller now anyway. Well, actually, I'm Professor Waller now. Obviously I had to have a picture of some shoes there. So I went to go be a teacher, and I started my PGCE in teaching, and this is Stoke High School in Ipswich, and I realised quite quickly that I'd made a very significant mistake, and I just needed to get out of Cambridge because it was an intense place, and just calm down for a bit. I think because Pip and I lived apart in Cambridge, and I like to think of him as he's my buffer, right? So I'm a little bit extreme, and then he calms me down. So I was without Pip for a while, and actually I didn't really, that was not a good decision for me. So I dropped out of teacher training, like, I'm gonna have to do this, gonna have to drop out. And then because I had a 0 hours contract with the library, I went back to work at the library. And in my lunch hour on jobs.ac.uk. And at the end of 2009, I saw this job. It was like just before Christmas. I'm sure it was like the 18th of December. And there was this job advertised at UEA as a senior demonstrator. I'm like, I can do that. I can definitely do that. And it had 2 weeks. So I'm like, who's going to apply for a job this time of the year, right? Like, that's ridiculous. Anyway, I applied for the job because I thought I could do it. And then I got an interview as a senior demonstrator in medicinal chemistry with Duncan. And I got a lovely call from Duncan saying, we didn't intend to appoint an external candidate to this position. I was like, yeah, makes sense now. But you blew us away, so we have to offer you the job. I was like, wow, thank you very much, Duncan. I'm very, very happy to hear that. And then I became integrated at UEA. I made really good friends with Paul and Leslie, who are here today. And Mark, who was the head of section at the time, who was my mentor and essentially told me what I had to do. He later became head of school after Duncan left. And part of what I did as a senior demonstrator was like, I did a lot of adding up, which was checking student numbers, which was also didn't really go well for me. And sometimes, especially, we needed to do it in a rush to split papers apart. And yeah, it was, anyway, I had fun. I also did some research on the side, and I was actually supervising students in Sue's lab. So special ed, Sue doesn't like having a photo taken, so I've just popped her in there. I have no photos of Sue at this time, but I was essentially supervising her students in the lab. And then, because that was a short-term contract, I worked for HFL Sports Science as a scientist, and I did a lot of screening. But I was quite lucky in the sense that a position as in chemical biology came up at UEA, and it was a junior position, so at the same level as a postdoc, but independent. And I applied for that, and I was lucky to be able to get the opportunity to start at UEA. Again, Duncan interviewed me for this, and he did say, actually, when he offered me the job, he says, we're taking a bit of a punt on you, so don't let us down. I was like, okay. So I tried my hardest. This is my first office, which I shared with Min, who is here today. What did I work on? I decided that I would go back and work on DNA structure, but I'd focus on something that I knew Shankar wasn't working on, because obviously Shankar was like one of the top dogs working on G-quadruplex. There was nothing that I could do that was going to be able to be as good as that. So I decided I'd work on i-motif, because I thought, well, I could just apply everything I know about quadruplex to i-motif. And then I made a lot of mistakes along the way. But this structure is acid, it's acid stabilized. So as a bit of an introduction to i-motif, this is a structure that can change the shape based on pH. And Shankar's group showed that you could make nanomachines using acid and base. And work by Lawrence Hurley, actually at the end of my PhD, showed that these structures, although they're acid stabilized, they could form under physiological conditions. And separately, I know that some people that were working for Shankar at the time also saw that these were present and under neutral conditions using single molecule work. So I felt confident that it wasn't completely idiotic to work on the i-motif. So I decided I wanted to focus on targeting i-motif DNA, and this is the structure that I now know and love. I graduated from Cambridge a couple of years later because I wanted to wait for the people that I was to graduate with. So I still see both Chloe and Keith quite regularly. Mum was able to join for graduation, so I was really, really happy that she was able to be there that day. I know what you're all thinking, this is all rather boring. What are you wearing? But I've forgotten about this. But Peterhouse had a 2-page document of what you can and cannot wear to graduation, including shoes must be of black and sober style, and also hair colour, exaggerated hairstyles and aren't inappropriate. So I couldn't graduate today, that's for sure. So I built my craft after that. So I did a lot of teaching, I did a lot of engaging with, I mean, the staff at UEA was super I was really happy working there. And I built up my group, I got funding, and so I got a small grant from the Royal Society. At this point, I was only on a fixed term contract, but in 2013, that got sorted out, so it enabled me to be able to submit for bigger grants. I had my first independent paper in 2013, which was showing that silver cations can stabilize these i-motifs, so you didn't need acid to stabilize them, and you can switch that back and forth with cysteine, and that was Henry and Camille that helped me work on that. And this is one of the projects that I did myself mostly. Leslie and Paul, who were my friends at the time, thought a great birthday present would be give Zoe a pattern of DNA and she can crochet her own model, which is great. And I've got here, this is, so Sue took this to the Southeast Asia and she decided it needed names. So this is Daphne the DNA. And unfortunately, you might notice, Mike notices, it's left-handed. And I only realised when I started putting it together, and I was like, I've done it now. So I've flipped this image. So, and then later on, it was like the most boring thing to make, just the same stitch again and again and again. So I'm not going to make another one. I haven't made a quadruplex yet, but I have made Iggy the i-motif. I should make a quadruplex. And then this is a DNA binding ligand, which can go in there. So I take that into schools. Thank you to Leslie and Paul for encouraging me. I also wrote a really nice review. I thought it was a nice review with Henry, and we had some really lovely feedback from Lawrence Hurley, who's one of the big people in the field. And I was really grateful to be able to host him at UEA together with Mark at the time. So he gave some good feedback on that. I'm grateful. And it was an odd time, 2014, there was it was quite a tough time for me. And that was the year also that Mum died, and she was ill for a long time, but it sort of hit me like a train when we lost her. And I got this email, like literally like a month after she died. And it was the type of thing that it was so bittersweet because I felt absolutely awful. But then I got my first major grant from the UCL, so I was super happy. And also that year, I got a little grant from Diabetes UK, which, as Cate has mentioned, I built on from there, and they gave me further funding later on. So after that, I was promoted to, rather than junior lecturer, to be a lecturer in chemical biology. And the year after, found myself pregnant somehow. But also I got the opportunity to become director of admissions for the M-Pharm at pharmacy. So I ended up doing lots of going around, traveling around the UK, giving lots of talks, which I actually really enjoyed. And sometimes, especially when I was pregnant, people didn't trust me to not drive into the side of the road because I was so tired. So he also came on some of those trips as well. In 2015, I did some work to further some of this, the work that we were looking at different types of conditions that could stabilize i-motif. And we showed that you could use, spoiler, we showed that we could use copper cations to change the shape of DNA from i-motif to a hairpin, which is a different structure, and back again. And we had help from a long-term collaborator and great friend of mine, Andy Gates. In addition to this, I found out that I have dermatographia, which is basically I can draw on my skin and it will kind of puff up. And I started doing Molecule of the Day on Twitter, and then that kind of got a bit of attention, ended up being in Daily Mail. And so somebody came and said, I'd like to write an article, can I take a few photos? And then like a week later, it was all in every newspaper. It was not what I was expecting, but it did lead to some other things which were quite nice. I ended up writing an article for The Independent, which was nice for me to be able to do. I had been approached by artists, and one of those is Fragile Self, and I think Jonathan and Anil here, so we did. So this is an image still taken from their music video, which was released earlier this year, and I can give you more details if you would like to see that, but. So Jonathan did all of the different writing on me as part of that, and I was the model. And I also ended up getting asked by the Wellcome Trust. So some of these images are now in the Wellcome Trust Wellcome collection. And although you can't see it there, I was quite pregnant at the time. And then this is the first total synthesis of a natural product. And that's Eddie, who is here today. And when I returned from maternity leave, I was had a really great opportunity. So Mark Searcy was stepping down as a Royal Society of Chemistry degree accreditor. And I got offered the position, so I took that. So I started doing degree accreditations for the Royal Society of Chemistry, alongside some of the other things I already did. So I worked for the local section. I also a part of the chemical biology and bioorganic group, and until recently also the nucleic acids group. We did a lot of finding different compounds to target i-motif structures, working with different collaborators. And in 2017, I had a paper that's one of my most highly cited papers that showed that actually i-motifs, you could just get some that were stable and they're all present across the genome. They worked together with Julian for that. And then in 2017, I got promoted to be a senior lecturer in chemical biology. And my group was so sweet at the time. They bought me this little like crown, which I do still have. It's on my office now. But I was really, my group are like always been super supportive. And I feel like it's a dynamic exchange. We both help each other. That year I also became a fellow of the Royal Society of Chemistry. We also do lots of things like Norwich Science Festival. So we did a DNA one in 2017, then pregnant again. I think I was doing a pharmacy one here, and then I had this really great idea that we could think about where can you put all of these different medicines and where they go in. So I had like Velcro all over my body when people could like, there was not suppositories, just to be clear. I know where everything's going with this. We had like Velcro on some of this, and you could see this like anyway, so I had a lot of fun doing that. And then I got further funding with Andy for a project in 2018, and also a project funded by Diabetes UK, and also became chartered chemist. And we did further work looking at how copper could change the shape of DNA, and working with Andy, we could show that you could change the shape of DNA from unfolded to i-motif, and then going back and forth to the hairpin as well. We also looked at how specific quadruplex ligands actually also interacted with i-motif. Franklin came along, so this is total synthesis' second natural product. And then following my maternity leave, I was taken off admissions and I became Deputy Director of Learning and Teaching. I did a lot of travelling with Sue and Paul, and we did some super interesting active learning workshops in Vietnam. COVID happened, and it's a dark time. I was so stressed, but also an opportunity came up to come to UCL. I did get it was an email saying, I'd like to have a chat with you. I was like, okay, yeah, let's have a chat. So I applied for this position as Associate Professor in Drug Discovery and got the job, and arrived here in 2020 as an Associate Professor. And in that year, we also had some nice papers. One of my collaborators is John Fielden, and we showed that we could identify using a light-up mechanism binding i-motif. And Phil, who worked on that, is also here today, and also worked with Nicole Smith looking at the interdependence between these quadruplexes and i-motifs in cells. I also do a lot of outreach, as Cate mentioned, and I did Soapbox Science. So in 2021, it was online, and one of my mentors, had advised that I go for the Associate Director for Research position within the school, and I was super excited to be able to do that. And then literally after I'd agreed to do that job, I found out I was pregnant again. Total synthesis of a third natural product, Griffin, and with a bit of help from Moderna. And I'd agreed to write a book chapter for Naoki Sugimoto, and it was one of the things I didn't manage to do before I went on maternity leave. So I ended up writing this book chapter. People were like, this is a lovely book chapter. I literally wrote it on my mobile phone whilst breastfeeding Griffin. So thank you to Griffin for your help with this. I should have put in this acknowledgement, actually. And I did soapbox science in person, and I made this lovely lab coat. Oh, I meant to bring it here. Oh well, you can see it. And my group are super supportive. This is us at the beach. And then last year, my application to become Professor of Drug Discovery went through. I was really happy, and I'm really happy to support my group and the work that we do. Last year also, rather than just becoming an accreditor, I'm now actually the chair of the Royal Society of Chemistry Accreditation. And then because of that, I'm now stepping down from my other roles at the RSC, and we've got the next generation here. I say Mike Booth's going to be taking it, has taken over as secretary of the Nucleic Acids Group. But my service was recognised as well as my work last year as a lectureship award from the Royal Society of Chemistry, Chemical Biology and Bioorganic Group, and I received that earlier this year. Other work, these aren't all my papers, but these are ones that I feel like are quite worth talking about. I have another long-term collaborator, Yilian Ding at the JIC, and we've developed a web server for trying predicting i-motifs in sequences and then building on that using machine learning. And then we also have been collaborating with Stephen Needle. I'm so excited. Using this compound that's actually in clinical trials and shows that actually this compound unfolds i-motif DNA, even though it also it stabilizes quadruplex as well. And Efrosini here drew these pictures based on the pictures that I drew for my PhD thesis. That's quite nice. It was very beautiful. It was the front cover in all of the journals that we submitted to. And I really am super pleased and proud to be part of SOP because there was a very rich history in working on nucleic acid structure and function. And I was very happy to be able to work with Gary and Joseph on this work, and basically combining some of the work that Chris Morris and I have been doing on DNA structure in the insulin gene, and then actually having a better understanding of the structure, both through crystallography, computational biophysics, and then also using an MR with Chris as well. So I very much enjoy collaborating with people. And then, yeah, last year I decided to go to be head of research department with PBC, and I've just started that job really. I'm still kind of winding down my associate director of research, but I'm looking forward to all of the excitement and the opportunities that it brings. And then finally, my final paper that I want to mention is that this paper, which is basically trying to find compounds that can modulate this particular gene. Emily she left the group in 2018. So I've got a bit of a backlog, and I do feel like moving and 3 children has held me back in some aspects. So people say, okay, I don't know how you do it all. I'm like, I don't. Some stuff's taken a long time. So sorry to Emily. But we did get it out. So what's next, apart from publishing more stuff that I should have published 5 years ago? I'm the type of person that's an opportunistic person. I like to see opportunities and be able to see them and see what can happen. So I don't know what's next, but I'd like to be able to be a good role model for others and show that that you can do both good teaching, good research, and good administration, and get that balance, and also have a family. But being realistic about it, that there are some things that are going to happen. You can't do everything. You're not a machine. And that just leads me to kind of thank all of my collaborators, all of my mentors, and there are so many. And I'm so grateful to so many different people, but I've tried to mention them as I've gone along as well. My colleagues at SOP, and there's so many inspirational people that work here, so many people that it's such a great environment. There's so much diversity here, and I was a bit worried about moving in the pandemic because I didn't know what moving meant. I didn't know when my lab was going to move. I didn't know whether I couldn't visit people, so it was quite a leap to make. It's quite a difficult decision, and I worry about a lot of things, as you probably all know. I was quite worried about what the people would be like because people at UEA were so kind to me. And but it's been amazing. I'm absolutely so grateful to be here and have the people at SOP as my colleagues. And the bit of humanity that in what we do, whether it be getting stuck in the middle of nowhere with Andy on the way to Brno, whether it's the Tweed run, whether it's my group finding it funny to write Zoe Smells on the beach, or one of your mentors coming to visit you, and then your children thinking that she's like an extra grandma. It's been lovely, and throughout I've always been very clear about what I am. Like I am a mother. I'm a mother of 3 children, and my group and my colleagues have always been so happy and receptive to kind of accept them as part of my life, especially my group, especially Effie. Is Griffin going to be here today? He is here today. So, and also just want to make sure that I acknowledge and thank my family. You've seen pictures of them so far, and yeah, and some of them aren't able to be here today, but I want to thank them for their support. And then also Pip and the boys. So Pip, as I said, is my buffer, and I saw someone on TikTok, oh, I can't believe I just said that, say that behind every great man is a great woman, and behind every great woman is a man that does the washing. And it's true, like, so he does more than the washing, like, so, but I don't do everything. I am very lucky to have a partner in life that shares responsibility and everything that comes with it and is supportive. So thank you to Pip for being my buffer and everything else. And I worried a lot for a number of days about forgetting things and important people. So if I have forgotten you, then I have worried for 3 days already about it. I've committed a lot of time to you, but thank you so much for your kind attention, and thank you so much for supporting me today. I very much appreciate it. Thank you.
Traditional that you don't get to ask any questions at an inaugural lecture. Sorry to those people online and also that, but we also do have the opportunity to ask Zoe those important questions, not only about the papers, not only about her external engagement, but also when did you get the amazing outfit? You will join us in the square now for refreshments. And so thank you very much for all coming. Thank you so much, Zoe, for a stellar performance. What an inaugural should be, not only about the science, not only about the education, but also your journey that brought you here. And we're all really pleased that you did choose the UCL School of Pharmacy. Well done. Thank you.